In today's video, I'd like to discuss cancer, what I believe causes it, and things we can do to work with our own surveillance system. Western medicine believes that cancer is caused by mutations in cells, typically progenitor cells, which are halfway between stem cells and normal cells, and that after 6 or 7 mutations, a cell can become cancerous. Yes, radiation, chemicals in the environment, these can cause mutations in cells and can lead to cancer. So indeed, cleaning chemicals out of the body is very important. There are other ways in which cells can become cancerous. One is by not having enough oxygen. Normal cells use oxygen to burn carbohydrates, fats, and proteins in the Krebs cycle. But if there's insufficient oxygen, a cell can then go into glycolysis, which is the burning of sugar in the absence of oxygen. This is one of the hallmarks of a cancer cell. Raising the oxygen is important for someone dealing with this condition. But once a cell has become glycolytic, it may become irreversible. So adding more oxygen may not be enough at that point. Another way in which cells can make that transition is if they're in too much of an acidic environment. Sometimes that acidity is caused from lactic acid, from the glycolysis, from the low oxygen. Sometimes that acidity is from diet or other issues. What the acidity does is it can trigger microtumors to become invasive. And let me explain what I mean by this. When autopsies are done on otherwise healthy people who die in car crashes, what's found is that half of the men and women have prostate microtumors and breast microtumors, respectively. A microtumor is something less than half a cubic centimeter that does not have its own blood supply and does not become invasive. You might consider it a small wart, harmless. But in the presence of acidity, these microtumors can be triggered to grow their own blood vessels and then become invasive and metastasize. So a protocol for this might be juicing. But again, manipulating the pH, while fantastic at keeping precancerous cells from making the complete transition, I don't know that that would be enough once a cell has become completely cancerous to turn that off. The third way or fourth way in which cells can become cancerous is a toxic environment, but this isn't a toxic environment from a chemical coming from the outside directly. This is from toxins the body makes itself. And to understand this, we're going to need to talk about how cells regulate their own systems. Cells have a very particular requirement for their environment. They want high potassium, low sodium, low calcium. They want toxins out. They want nutrition in. And the way they accomplish this is with pumps, transporters, and channels. Pumps use energy, ATP, to push something in or out of the cell against a pressure gradient. Transporters come in 2 types, symporters and antiporters, and they use they'll move 2 things at the same time. So let's say there's something inside the cell that the cell wants to remove, and there's something else inside the cell that wants to go out on its own volition, just a pressure gradient. The symporter will grab both of them and use the motive force of one to pull them both out. An antiporter is more like a revolving door, where if there's something on one side of the cell that the cell wants to bring in or get rid of, and on the other side that wants to move across of its own volition, it'll connect those 2 and swap positions. And finally, you've got channels, which are like open doors. They're not very particular, and lots of things go through quickly. The way cells work is they'll use the channels for mass movement, and then fine-tune what they need with the transporters and the pumps. What ends up happening as we're exposed to metals is they jam up the pumps, transporters, and channels so that nutrition can't get in, toxins can't get out, and the cell finds itself in a more and more toxic, less and less hospitable environment. And its adaptation to that gradually toxic and dysfunctional environment is cancer. So on the one hand, we have external events like chemicals or radiation causing some kind of random damage to the DNA that makes it so the cell cannot function normally. And on the other side, we have toxic metals that slowly dysregulate the cell's ability to manage its own homeostasis, and the cell adapts to that, and that adaptation is cancer. Again, there are ways in which you can deal with some of these things. There's oxidative therapies, there's hyperbaric oxygen, flow oxygen, increasing the circulation. There's juicing for acidity. There would be a ketogenic diet to rob the tumor of its sugar. But remember, the liver makes sugar, so we can never fully stop that. But if we're going to get it at the base cause of these things, we're going to have to remove the chemicals and the metals which are causing all these problems in the first place. So it's important to have some kind of protocol for dealing with chemical toxicity. We've already talked about Xenoplex in other videos. That's one thing you might consider if you're concerned about chemicals. Let's talk about how the metals specifically cause problems. The issue is called molecular mimicry, and that's when one metal looks very much like another. As an example, both mercury and sodium have a size of 1.02 angstroms, so they're basically identical in size. But divalent mercury has a positive 2 charge, sodium a positive 1. So when the sodium channel or pump or transporter grabs on to mercury by mistake, it may not be able to let go very easily. Imagine that you're in a busy department store, and then somebody just sits down in the middle of the revolving door and doesn't let anybody get in or out. It's going to cause problems, right? Same thing with barium and potassium. Barium is 1.35 angstroms, potassium is 1.38, almost identical, but barium has a +2 charge, potassium +1. So if the barium gets stuck in a potassium transporter or channel or pump, it might not let go very easily. So what happens as these pumps, transporters, and channels fail? Well, the potassium inside the cell starts coming out, the calcium goes in, and this loss of differential causes the voltage on the cell to drop, like the battery in your cell phone losing charge. Now, normal cells have about -38 to -93 millivolts when measured. That's their reading. Prostate cells are a little lower, which is why they're more likely to get cancer than other parts of the body. But as a general rule, -38 to -93. Now, if you take a cell and drop its voltage, you can make it cancerous. On the other hand, if you take a cancer cell and raise its voltage, you could stop its growth. If you take a cancer cell to -70, it'll stop growing. And this is the way in which things like multi-wave oscillators and electrical therapeutic machines work. They temporarily raise the voltage. But again, this is not really getting at the cause of the problem. It's temporary. The cause is that the membranes themselves have jammed up pumps, transporters, and channels and cannot create a proper voltage. Just giving it a surface charge for a little while in the machine, as great as it is, isn't really getting to the issue. So let's talk about what Western medicine has to offer. Chemotherapy came after experiments with mustard gas and came after penicillin. Now, penicillin was a lifesaver, and it works very quickly. If you put a little penicillin in a petri dish, within a day, the bacteria is already starting to die, if not dead. So when people were looking to research how to kill cancer, they had the same mindset as penicillin. They said, well, we want whatever we're going to test against cancer to kill it in a day or two. So they took lots of compounds from the world and put them in dishes with cancer cells and waited to see a day or two if the cancer cell died. If it didn't die or didn't significantly start affecting the cancer in a short period of time, they said, move along, let's find another compound. But that's a kind of selection bias, because we're not dealing with an acute infection like a bacteria. We're dealing with a chronic situation. And chronic situations, sometimes you have to go out the same way you came in. Yeah? It's not a quick fix. So all the natural products that might have showed results on day 5, day 10, they never got tested. They weren't fast enough. But those are really where we want to go to. Because the things that are powerful enough to kill a cancer cell in a day or two are also probably powerful enough to kill us or hurt us, because the cancer is us. It came out of our own tissue. It's not a foreigner. Yeah. One of the things that is difficult with dealing with cancer is its multi-drug resistance. Now, there is something called Pglycoprotein. It's the general detoxifier of the cell. And as a cell is being exposed to more and more toxic environment, whether it's chemicals or a toxic internal environment because its pumps and transporters and channels are getting blocked, it upregulates the Pglycoprotein. And in the 5 years or so it takes for a cancer to grow large enough to be visible and palpable, it's making more and more of these Pglycoproteins and being exposed to more and more toxins. So by the time a person is able to notice a tumor, it's got a lot of Pglycoprotein on it. And then so cancer cells are basically the most toxic resistant cells in the human body. They have the greatest capacity for detoxifying because they've been doing just that for the last 5 years. This is why chemotherapy is so difficult, because when researchers are trying new chemotherapy agents, they put it in the cancer and the cancer just dumps it back out again. That's the Pglycoprotein. So if we're going to use that model, a chemotherapy model, to try to work with cancer, it can't be something that the Pglycoprotein will see as a toxin. When I see clients, one of the things I'm researching is mixing zinc with ammonia. Ammonia is very tasty to cancer. It has learned to use the toxin ammonia, which we normally get rid of, as a fuel source. Now, you know, if you put nitrogen on your plants, they grow very quickly. Well, ammonia, nitrogen, is what makes cancer cells grow very rapidly. And in a dysfunctional cell in which the ammonia is building up, the cancer cell has learned to use that ammonia as a fuel source. Now remember, cancer cells are not very good at moving metals back and forth. They've got those things jammed up, those pathways. So it's possible that if we can attach zinc to ammonia and have the cancer gobble up the ammonia, it may end up getting so overloaded with the zinc it can't get rid of that it makes it challenging for the cancer. What can we do on a day-to-day basis, though, right? So in trying to figure out how I wanted to protect myself and my family and my loved ones, I looked into the animal kingdom. Now, with heart disease, humans are one of four mammals that don't make vitamin C. And so there's very few animals that get heart disease. But most animals get cancer. So there's a great resource there to trying to understand, to try to see how other animals have developed defenses against it. So let's talk about 3 animals that I believe have fantastic defenses against cancer. The first is the elephant. Elephant lives a long time, is much larger than us, both of which are things that tend to make cancer more likely, and yet it has a fraction of the cancer we have. Why is it? There's a gene called the TP53 gene that is one of the main genes that the body uses to deal with cancer. And what it does is it finds a cell that looks a little funny, and it stops it, it freezes it, that's called cell arrest. Then it will fix it if it can, and kill it, apoptosis if it can't. That's T53. Half of human cancers are associated with a failure of our T53 gene. We've only got one gene. If that gene gets damaged, there goes our main surveillance system, yeah? Elephants have 20 copies of the TP53 gene. So even if they lose one or two, they got 18 more. I believe this is what gives them their fantastic resistance to cancer. So how might we be more like elephants? Well, ellagic acid, something found in the plant kingdom, you can find it in pomegranates, red raspberries, and the seeds, lots of plants, acts like TP53. It causes cell arrest. It can repair damaged DNA or trigger the repair, and it can stimulate apoptosis when necessary. So I consider ellagic acid an external TP53 analog. Not only would I take it if my own TP53 wasn't functioning, but even if it was and I wanted more, if I wanted to be more like the elephant, ellagic acid, I believe, gives us that. Another animal we can look at in nature is the ant. Ants don't get cancer, and you could argue that it's because they don't live a long time, but they have a very remarkable immune response. They don't have an immune system. What they have is an extraordinarily powerful immune genetic repair system. Basically, they say, I don't care what bug I get, I'll fix myself so fast, it just doesn't matter. They do this with an aldehyde called iridoidal, which was made famous by the famous German doctor, physician, scientist Hans Nieper. So one day, it will be great if iridoidal will be produced by the metric ton and available by the spoonful. Until that day, the best way to get iridoidal is to eat ant extract. And I do that every day. It's ground up. It tastes a little bit like coffee. I don't really notice it, other than maybe the energy boost I get from it. And that way, I believe I'm imparting the genetic repair mechanism that the ant has into my own system. The third animal I want to discuss is the naked mole rat. Now, this spends its life underground, it's hairless, and one of the interesting things about it is it lives 7 times longer than a mouse of the same size. And it is immune to cancer. If you try to give a naked mole rat cancer in a lab, it just won't take. Well, that's a neat trick. I'd like to learn that trick. How do you do it, naked mole rat? Well, one of the things we know is that it creates a lot more of hyaluronic acid than we do, which is the connective tissue, and it's a much thicker hyaluronic acid. And it doesn't secrete as much of the enzyme, hyaluronidase, that breaks down this hyaluronic acid. So they have a very strong connective tissue. Now, I don't think the naked mole rat can't get cancer. I think it can't get invasive cancer. I think what happens is it gets microtumors like all animals, but its connective tissue is so strong that it just can't invade and go anywhere. Hyaluronidase, the enzyme that breaks it down, is also the same enzyme found in spider and snake venoms. So what could I do to be more like the naked mole rat? Well, I eat a lot of hyaluronic acid. I want more of that in my system. But I also take things that suppress or downregulate my hyaluronic acid enzyme system. So amla will do this, so will fat-soluble vitamin C. So these are the 3 strategies I think are important to understand when dealing with this. One, we want to be free of toxins, free of metals which are jamming up the pumps, the transporters, and the channels, free of chemicals that are causing the mutations of the DNA, and have enough circulation that oxygen gets in, maybe go on a ketogenic diet a couple of days a week and give the body a break from so many sugars. I think that's a very important baseline of things to do. The second thing, I think, is it's really important to have a very aggressive surveillance system so that those microtumors that we are prone to stay as tiny little harmless warts and don't bother us. And for that, I look towards the elephant, the ant, and the naked mole rat. And then finally, I think it's good to have someone on your side that has a very powerful and aggressive protocol for dealing with cancers that have become invasive and that need something with a bit more kick once that's come down the line. In order for me to become more like the elephant, the ant, and the naked bull rat, personally, I take large amounts of ellagic acid, which I believe is a TP53 analog, ant extract, hyaluronic acid, and hyaluronidase inhibitors, along with a few other things that work with it. And if you're interested in experimenting with those, we make a product called mitopanol that has those ingredients in it. I wish you well.